ready, but I am.
So 37, just a second. So I just posted a Slack message to everyone. It will go up a tiny bit with the numbers.
Okay.
At the very beginning, we didn't know how many people will actually join for each session, so we didn't know like how long we should wait. But now it like kind of converged to about 50. So when we reach 50, we're taking off.
50?
Yeah. So there are like 10 more people joining. So right now we're at 40.
Oh, there you go. Okay, that makes sense.
It's like if you have a free workshop that is virtual, you cannot ever tell how many people will actually come, because for them canceling is free, just like the registration was. And
yeah, that's fine. Yeah, I mean that's a good thing about online. You don't have room capacity. You can't do it.
Yeah, I actually think that we've done really well. We originally wanted about 50 people, and we allowed 70, and in the end 50 came. So
nice. Hopefully my tutorials won't lose half of your audience.
Oh well, so far you're on like nearly all, so like you are on 85%. Okay. I suspect that a few more will join in a second, but I think we can just start. Would you like an introduction, or would you like to introduce yourself?
Well, either way, I'll introduce myself. Why not? So should I go ahead and get started? Okay. Let me share again then. So, let me just say hi to everyone before I share. So my name is Siavash. So that's where the difficulties start. My name is hard to pronounce. You guys have to get used to it. Sorry about that. Let me start sharing. Maybe it helps if you see my my name. I am at University of California, San Diego. This is kind of where our campus is in the background right there. I'm in the electrical and computer engineering department. And what I do is basically develop bioinformatics algorithms. That's what my lab focuses on. A lot of our work is related to phylogenetics, but also we work on ways to use Skmers for sort of sample identification and questions in ecology. And that's what I will be talking about today. Okay, so I have a presentation and I have tutorials. I'm going to switch back and forth between them. So I'll start with the presentation. And then in the middle of it, we will switch to the proposal, and then at some point we will switch back. So we'll keep going back and forth, so that no one gets tired of me just speaking. Okay, so I'm guessing all of you are biologists. So by the way, I'm not a biologist by training. I'm a computer scientist, but I've picked up a little bit of biology here and there. Let me get the chat open as well in case. So during the talk, feel free to ask questions in the chat. I'll try to monitor it, or just like unmute yourself or raise your hand, or whatever the procedure is. But feel free to interrupt me. Okay, so you guys are biologists. You know this better. There are a lot of questions in ecology and evolution, but also in sort of conservation, where it would be really helpful if you were able to identify what a sample is, what exact species it is, what exact population it is, and also if you could look at the sample and know something about its population, size, and things like that, it would be very helpful. Now, for identification itself, the method that is the standard these days, I would say, is barcoding, right? We have these little barcode genes like CO1, or depending on what organism you have, there are different markers. CO1, I think, is the most well-known one. These are basically just one gene that is supposed to have enough signal to be able to tell apart the species, but it's like cheap enough to just it's actually very cheap to just sequence this one gene using PCR. So you have your sample, you get the DNA for this particular barcode gene, and you search it against a reference database like Bold, and you figure out what you have as much as possible. Now, there are some limitations here. One of the limitations is that because this is just one gene, you can have limited resolution. So oftentimes you can tell what genus something is from, but not exactly which species in that genus you have. And this is a well-known thing. So, I guess 5, 6, 7 years ago, at this point, people thought, well, whole genome sequencing is getting cheap. Why can't we just use that for identifying a species? Now, the problem is, if you want to sort of get a genome, get enough coverage of the genome. So, the way you get full genomes, of course, is you get high coverage of the genome. You get so many reads so that every part of the genome is covered, let's say, at least 30 times. And if you have that much data, then you can assemble the reads into a genome. That is still expensive. Expensive not as in if you have one species and you're interested in, you can of course do it, but you can't do it for every single sample that you collect in the field. That would be too expensive. So, who knows how many species are out there, but, and this slide is quite outdated now, but the point is the number of genomes that are assembled are always is going to be still quite a bit lower than what is out there in terms of species that exist. So one alternative to full genome, sort of assembling full genomes, is genome skimming. It's been talked about since 2012, so that's about 10 years now. The way it works usually is that basically you get low coverage data across the genome. So let's say 1x, meaning that you get enough reads to cover your genome at 1x. So that's not nearly enough to get any kind of assembly. It's not close to enough for assembly. But what can you do with it? Well, people who came up with genome skimming, for example, these early papers that I am citing down here, what they suggested you would do is take advantage of the fact that organelle genomes are overrepresented. So what that means is that if you have 1x coverage, or what I'm drawing here is more like half x coverage, or let's say this is your nuclear genome, you have something like half x coverage is what I'm showing here in red. Organelle genomes, they're going to be overrepresented, so that you might get like 4x, 5x coverage for organelle genome, and that's of course because you have multiple of the organelles. So if you could, if you could get the organelle genome assembled, then you could do your identification of species using that. That's still better than one gene, right? It's a genome is still better than using just one gene. That was the idea. Now, what about the nuclear genes? Can we take advantage of those? Because it's true that organelle genes are overrepresented, but the organelle genome is still much smaller than your nuclear genome, right? So the total number of reads that you have from organelle genome is going to be a tiny fraction of the total number of reads. So when people do this standard thing with genome skims, you're still getting rid of 99% of the reads. You're just using the 1% that happens to be from organelle genomes and is overrepresented. Okay, so what that seems like a little bit wasteful. Get rid of 99% of reads and keep the 1%. The question is, 99% because it's low coverage. Is there anything useful we can do with it? We can't do assembly because it's low coverage. And a lot of times you do not have, so another idea might be, okay, so why don't we map these to all the reference genomes? Mapping isn't just like align these reads to reference genome using a bow tie or Minimap 2 or something like that. And whatever genome it aligns to, you identify that. That would work if you have a reference genome from every single species of interest. But oftentimes you don't, right? A lot of the reference genomes you are interested in, they don't have reference genomes. So in that scenario, what do you do? Well, that's where Skmers come to the picture. So the idea here is that we are going to try to use what I call the bag of reads, right? So just a collection of reads that you have, let's call it a bag of reads. We want to use our bag of reads as the barcode of the species. So the barcode itself, the bag of reads that you have no assembly, nothing, that bag of reads is your barcode. So for references, you are also going to have a bag of read. And for query, for what you are trying to identify, you also have a bag of read. Then what you essentially need to be able to do a species identification is a way to take two bag of reads and compute the distance between the genomes that has generated to those two bag of reads, right? So if I had the full genome assembled here and here, what would have been their distance? I don't have the genome, but I have these bag of reads. Can I at least estimate the distance between them? And of course, the difficulty here is that we want to estimate this distance, even though both of these have low coverage, right? And by low, we mean maybe 1x, maybe half x, maybe even 1/4 of an x. We don't mean like, I don't know, 1/100. Okay, so the question is, can we compute the distance between two bag of reads with low coverage? Any questions so far? Let's see if someone raises their hand, will I see it? Is everyone good? Okay, so moving on. So once, of course, you have these distances, a species identification is easy enough, right? So you have a bunch of reference bag of reads, you have a query bag of reads, you compute these distances, whichever is closer. If it's close enough, you identify it as that. And I'll get back to this question of identification later, but like conceptually, you can see where we are going with this. Okay, so what do I mean by distance? By distance, I basically just mean Hamming distance. So let's assume you have two genomes that are perfectly aligned with each other. We don't have this, but assume we had the genomes aligned. Then you would just look across and see what percentage of the positions are different. So here I'm drawing 16 positions, 2 of them are different. So I say the distance is 2/16. So that's just Hamming distance. We call it sometimes GND, genomic nucleotide distance. I'll just say distance from now on, right? I don't want to keep repeating Hamming distance. Okay, so that's how we define distance. Now, how do we compute it? The idea, of course, is to use Skmer representation. By now, you all know what a Skmer is, so I won't repeat this. You have but we are going to extract Skmers from reads, right? So each read is 150 base pair. We are going to divide it into smaller Skmers. I'm showing 7 mers here. In reality, just so that you know, in the tools that I'm like in all the applications that I'm going to describe, our Skmer length is going to be somewhere between, I don't know, let's say 21 to 31. That's kind of like the reasonable range for the K. We default to 31. But you can go down up to 21. But for the type of Skmer analysis I'm talking about, we are talking about long Skmers, not very short Skmers like 7 mers that I'm showing here. Okay, so that's that's what the Skmer is. Let's move on. Now, how do we use Skmers to compute distances? So let's say you have 2 genome skims. I'm showing 2 reads that were generated from to a specific part of the genomes where there were no mutations. Of course, when you look at the Skmers from each of those reads, those Skmers are identical between the two genome skims. Now imagine you have a read from a region of the genome when there was a mutation, right? So you have an A over here, you have a T over there. All the Skmers that sort of overlap with this one mutation, they all are going to be different to the between the two genome skims. So these 4 Skmers are going to be different, right? So if I just look at the set of Skmers on the bottom and look at a set of Skmers on the top, I'm going to see that like a bunch of them are identical, some of them are different. Can I just compute some measure of how similar the set of Skmers is? So each genome skim is going to be defined by its set of Skmers. So we started with a bag of reads. Now we are going to go to a bag of Skmers. Each genome skim is just a bag of Skmers. And so I have 2 bags, one from, let's say, the reference, another bag from the query. And I'm going to compute what's called the Jaccard Index. Jaccard Index, you can define it for any 2 sets. It's basically the size of the intersection, this region over here, divided by the size of the union, right? So you just take this, you just take this intersection region here, right? You divide it by the union, that's Jaccard Index. So if it's 1, it means your 2 sets are identical. If it's 0, it means the 2 sets are disjoint and everything in between, right? So we are going to use the Jaccard Index between this set of Skmers, right? So A1 and A2 in our case are going to be all the Skmers in the 2 samples, right? Some Skmers are shared, some Skmers are not shared. Index for them. Okay, so that's your Jaccard. How do we go from, so we can, okay, so the one important advance sort of like 5-6 years ago was that Adam Flippe's group published this tool called MASH, which a lot of you guys might have used already. MASH uses a sort of very well-known computer science technique called min-hash to compute this Jaccard index in a very efficient way, right? So very quickly, like without a lot of computational resources, without waiting a lot, it can compute this Jaccard index. It's actually like approximating it, but it approximates it very, very well. And it's very fast, right? So if you have 2 genomes, each of them represented as 1 gigabase per worth of reads, faster or slower, these numbers are with this slowest setting. It can become faster if needed. Okay, so we have MASH. With MASH, we can compute this Jaccard index easily, right? But Jaccard is not the same as distance. To compute the distance from Jaccard, a little bit of calculations are necessary. Of course, people who develop MASH also like notice this. This is not such a difficult calculation. We can go quickly look at it. It's not important if you don't understand it, but let's just quickly go through it. So let's say W is the number of Skmers that are shared in that region in between, and let L be the number of the total number of Skmers that we have in each of the genomes. For now, assume it's the same number of Skmers. Then Jaccard is going to be just W plus L minus W, right? The minus part, of course, is because the shared ones, you don't want to count them twice, right? So L is size of each of these circles, and W is this region in between. So the union is L plus L minus W, of course. Now, what is W? If the distance is D, for a Skmer to be the same between the 2 genomes, each of the K positions must be not changed, right? And remember, our definition of the distance was basically probability of each position changing, right? If the distance is 2/16, it means that that's the probability of any position changing. So if you make these kind of like randomness assumptions, if you assume everything is like completely uniformly random, then the probability that the Skmer is exactly the same between the two genomes is going to be given by this thing I circled over here, right? So you need each of the K position not to change, right? And you replace that, and from there is just like very simple math. You can compute what D is given the Jacquard. So if I have Jacquard, I can compute distance from it. I hope that's clear. Okay, so Jacquard and distance are very closely related to each other. The numbers are going to be like vastly different, but you can go from one to the other easily once you choose what the size of your Skmer is. Okay, so far so good, I hope. Let's move on. Now, does this approach work? So we took some insect genome. I forgot what. I think it was some sort of Anopheles. And we randomly added 5% mutations to it. So our distance, what we call D, is like 5% in this example, right, over here. So we have 5% differences. And what we are doing is we simulated two genome skims at 16x coverage between the two. And so we have the actual genome skim from this species. We have another simulated species that is 5% different. From both of them, we simulate genome skims, and then we use MASH to compute their distance. And when we have 16x coverage in our genome skim, this works beautifully, right? This is 5%, red is 5%, blue is kind of like right on top of it. So we get a very accurate estimate. The difficulty is when you go to lower coverage, by the time you get to like 4x, I think this is 4x. By the time you get to 4x coverage or lower, you see that we are overestimating the distance dramatically, right? If the distance is 5%, we could estimate it to be like 10% if we have 1x coverage. This is 1x. So that's not good, right? I mean, you don't want to overestimate your distance by a factor of 2. So this idea works only, so far it only works if you have high coverage, which makes it kind of like useless for us. We wanted to have like 1x coverage or something. But this problem can be rescued. So let's first figure out why it is that we have this problem. I want you to understand conceptually, and then I promise you in 5-10 minutes we switch to tutorials. Okay, so here's the reason. So let's say you have your two genomes and they are identical except for this one mutation when they are different. When you compute high coverage, when you have high coverage of the genome, all the reads coming from these blue regions are all the Skmers coming from all the reads coming from these blue regions are going to be shared, right? Because with high enough coverage, let's say 4x or higher, every single region in both genomes is almost certain to be covered at least once. Sorry, I missed the question. Yes, L was the size of the genome. Basically, the number of Skmers in the genomes. They're not necessarily equal. There's like a little fix that you can do for it. For that demonstration, I wanted them to be the same just to make the equations easier. But if they're not the same, you can sort of correct for that as well. It was for the previous slide. I missed it. Okay, so let's keep going. So what I was saying was that if you have 4x coverage or higher, all of the genome is going to be covered at least once with high probability. So every Skmer coming from every region that is not mutated is going to be shared. So all of the blue is going to be shared. All the Skmers coming from this kind of like black region are not going to be shared. Now, what happens when you have low coverage? Well, if you have low enough coverage, let's say even 1x, when you have 1x coverage, something like 30% of your genome is not covered at all. And of course, that's because some parts are covered multiple times, right? Now, let's say you have low coverage. What's going to happen is that some regions are going to be like this region over here. Some regions are going to be covered in one of the two genome schemes, but not the other. Look at this one right there. Well, Skmers in this read are not going to be shared between two genome schemes. But the fact that they are not shared is not because there is a mutation. It's because it wasn't covered in the other one, right? So that, but on the other hand, some Skmers, like Skmers over here, are not shared because there was a mutation. So there are now two reasons why Skmers might not be shared. One is lack of coverage, the other is mutations. And that is why we are overestimating distance, because cases like, because cases like this, because cases like the one over here are also going to be, like in that little equation that we had, it doesn't know it's because of lack of coverage, right? So it's going to assume this is also a mutation. So your distance is going to be overestimated dramatically. Okay, so let me see. Okay, so I don't have a slide on it, but you can also, I had a slide and I don't see it right now, but you can imagine, oh, there you go. I just had to skip it. Let me skip it. You can also sort of notice that if you have sequencing errors, the same exact kind of thing happens, right? So this is what we had without sequencing error. So now let's say we know that reads also have sequencing error, right? And now look at what happened with this Skmer. This Skmer is not shared, not because there is a real mutation, just because there was a sequencing error that happened on that base pair. If you have sequencing error, that also reduces the Jacquard, which means it increases your distance. So that's the second problem we have to worry about. Okay, how do we fix this issue? Well, we do another round of modeling, right? So coverage of the genome with shotgun sequences is random. Because it's random, we can assume that sort of the way it covers the genome is a Poisson process. Once you know it's a Poisson thing, you can easily sort of account for its impact. So what we came up was this more complex equation. This is similar to the equation we saw before. It's just a little bit more equation. So to Marillo, you notice that here we have L1 and L2. If you have two genome sizes that are different, you can kind of incorporate that. So you see these new parameters. You have eta and you have what was this? Psi. So these two parameters are basically functions of your coverage and sequencing error. Sequencing error we're going to show using epsilon. Coverage is lambda. So eta is just the probability that the Skmer is covered at least once in a genome scheme. So that's very much a sort of a function of coverage and error. And also we have the average number of Skmers per each locus, right? What's sort of something very similar to coverage as well. These two parameters, coverage and sequencing error levels, no one is going to tell us what they are. So we have to come up with a clever way to estimate them. But once we estimate them, this equation right there is going to translate your Jacquard and these parameters to the distance. So all we now need to do is to compute these parameters. How do we do that? Well, to compute those two parameters, we are again going to look at the Poisson. We are going to now also look at the Poisson distribution. Okay, there's a question. would not the net effect of sequencing errors increase the Jacquard? Yeah, so Jacquard is a similarity, is not a difference, right? So when you have sequencing error, it's going to increase the distance, which means decrease the Jacquard. So what you're saying is correct, Simon. It's going to end up making more differences. It's almost impossible, very, very unlikely. It's just that you were getting, I think, Jacquard wrong. Jacquard is similarity, not difference. Okay, so how do we compute coverage and error? Okay, so again, we look at the properties of Poisson distribution. Let's get, I think you all have learned about Skmer spectra already. So this is a Skmer spectrum. Basically, the x-axis is how many times you saw a particular Skmer, 1, 2, 3, up to 10. And y is how many Skmers were observed that many times, right? So if a Skmer was observed 10 times, it's going to go in this bucket. If when you do random sampling across the genome, like I said, the Skmer spectra is going to be a Poisson distribution with mode equal to the coverage. So if you see something like this distribution, you know that your coverage was 10x, around 10x, because your mode is around 10. Okay, so if you have high coverage, you can just look at this distribution, immediately you know what the coverage is. But what happens when you have low coverage? Let's say your coverage is 0.7x. Well, you won't have a mode because most of your Skmers are going to be observed exactly once. Now, if you had 1x, it would actually like visually look very similar to this. You would also have just one mode at 1x. So, so you can just, so you cannot just look at the mode, but we shouldn't despair. This distribution does change in subtle ways. Like you can't visually see it easily, but if I change this 0.7x coverage to 1x coverage, my mode is still at 1x, but all of these bins are going to shift a little bit. And that's what we utilize. You know, all of these, like what this histogram looks like is going to be a function of your coverage, genome length, and error rate. And we know what this function is. You know, we can model it mathematically using a Poisson model. For errors, we are going to assume they always sort of add to this bucket 1, always create sort of an unseen Skmer. The probability, if your error rate is low enough, like less than 1%, the probability that 2 Skmers will exactly create the same Skmer by error is low enough that you can ignore it. Okay, so we have a function that goes from lambda, epsilon, and sequence length to these figures. We observe the figure. We can kind of like optimize this function and find which parameters would give you a distribution that's closest to what you have observed. And that way you can sort of estimate all 3 of these parameters: length, coverage, and error. And that's how we do it. Okay, that's so just to summarize everything, you have you have a reference genome scheme, you have your query. You do 2 things. You look at this Skmer profile that I was just drawing, this sort of Poisson distributions. You get those Skmer profiles for both reference and query. For each of them, you compute your coverage and error rate and sequence length. On the other hand, what you need to do is do that sketching thing that I very briefly described what MASH does. And by sketching, you can compute Jacquard between the 2 genome schemes. Once you have Jacquards and estimates of the parameters, you kind of plug them in that equation that we have, and you get the distance. This process is what our method called a skimmer, which we are going to look at right now, implements. A skimmer just implements this process. We are going to have a little tutorial of it in a second. Before I go to the tutorial, I want to show you some results. Any questions before I jump to the results? Any questions? Okay, Seems we are good. So I'm gonna show you quickly some results. So remember this simulation I showed you when we had 5% error and their error was going up. We also did simulations with like 0% distance, 1/10 of a percent, 1%, all the way to 20%. You see the same pattern. When you use a skimmer with various coverages, even as low as 1/8, right? This is 1/8x coverage. Still, it works pretty well. When you get to very large distances, our method does have a little bit of bias, right? This is not just like random noise. We do have a little bit of bias around like 20%. And we go up to 20% because this methodology, it starts to fail once you get to 25% distance or higher, right? The maximum it can handle is like at most like 25, 26, somewhere around there. After that, it starts to fail. Because at that point, Jacquard is basically 0. Or very, like it's such a small number that you can't estimate it correctly. But I mean, this is not a huge bias, and it works pretty well with smaller distances. Okay, so that's the first results. Those were simulated pairs of genomes. These are pair of like actual genome skims that we created from like actual genomes. So like we took pairs of birds and computed their distance. We know that quote unquote true distance because we do have assemblies for these. And the assemblies are going to give us like the true distance, which I'm showing in red. And then from genome skims with low coverage, we can get the estimates. These are pairs of Drosophila genome skims. Again, the same thing. If you use a skimmer, the distances have very low error. The color here is error for all pairs of Drosophila. If you just use MASH, it's going to be higher here, or coverage is 1/2x. Okay, so before I get to this more interesting example, let me see what are the questions. Can't you obtain the error in the read data from Fred scores? No, I don't think so. So Fred scores, like their interpretation is very, very tricky. They do not really mean what percent, like there is no easy way to get from them an estimate of what percentage of the base pairs are erroneous. Fred scores means something more subtle. I don't want to get into what they actually mean because I also have to always think about it to remember it correctly, but they don't mean what we want. They have a kind of a different meaning. And also they don't give like you an actual number. They give you, they're just very hard to interpret. Have you checked how well it performs with closely related species? There you go. Sabrina, I can answer your question with the slide we have. So forget closely related species. By the way, if you're talking about closely related species, these are, for example, two eagles that are two eagles, right? Both from the same genus. Their true distance in red is very low, and this distance is calculated very, very well. But let's even go further. Let's try to compute distances between individuals from the same species. Two individuals from the same species. So we took this finch data set that was published a couple of years ago in Science. So I don't have the date here. They have a bunch of zebra finches here. They have other species as well. So in this figure, I'm focusing on zebra finch. Based on the assembly, they could compute theta. So I'm pretty sure everyone knows what theta is. Theta is just the average. It's basically just the distance that we compute, average over all pairs of individuals within a population. So they had a population of, what was it? I think 20, something like 20 finches. And they computed based on the assembly that the pairwise distance was 0.8%. So a little bit less than 1%. We used 2x coverage and estimated with a skimmer that distance, and we get 0.7. So that's pretty close. They also had like a population for long-tailed finch. The true quote unquote true distance was 0.55. We get 43. It's still pretty close, but not exactly correct. We have now come up with a new version of the skimmer that actually corrects that remaining error as well. So respect is a tool. If I have time tomorrow, I'm going to talk about respect. We just published it. The point of respect is to be able to model repeats in the genome. So I avoided talking about like actual repeats in the genomes so far because the Skimmer itself doesn't model them. And the repeats have a small, very small impact, but those is like a small impact that could actually mean something when the distances are very low, like here, right? You go from 82 to 0.7. But we now have a way to kind of estimate the repeat spectra from the genome skims as well and correct for that. And once you do that, your distances become very good. So this histogram here is showing sort of the average distance, the histogram of the pairwise distances, and their average is 83, what you want, and you get also like uncertainty estimates. So that I hope answered Sabrina's questions. Now here is a cool thing down here. So in this data set, they also had a bunch of like individuals that belong to the same family. So parents and children, and their distances is about half or less of like 2 average distances. And that's what you would expect, right? For diploid, even if you have like, a family would have much a smaller distance, and that's exactly what we see down here.
Can I ask a question on that?
Go ahead.
So would it be possible to go back to the same individual in a situation, for example, you're looking into getting some logging that someone cut from the forest, and then you want to see if that log is like illegal logging or legal. So you want to go trace back like a log that is on a truck somewhere, and then you want to see if it comes from the exact individual where the paperwork says it comes from. Do you see what I mean?
Yeah, so individual level, so you're trying to distinguish individuals within the same species. So this result here, so ideally, right, if you have the same individual, you get a distance that is very close to 0. That depends on ploidy still, right? Like even if you have identical, like the same individual with low coverage because of, let's say, diploid, because if you have 1X coverage, each read could come from one chromosome or the other. So there is a little bit of difficulty there, but hopefully the distance is going to be close to 0. We haven't actually tested that, so I hope it would work. It'd be a very nice thing to test. We are hoping to test that at some point. This distribution gives me some hope, right? When you get 2 unrelated individuals, you get distances that are large. When you get to members of the same family, distances become a lot smaller. And hopefully, if you compare 2 skims from the same individuals, it's also lower than this. You might not be able to distinguish like members of the same family versus something to itself, because those distances might all be like so small, but at least you can get close to that resolution.
Okay, thank you.
No problem. Another question, am I correct to say respect uses repeats to get individual differences? Not exactly. So it's not using repeats to get to individual differences. It's something a little bit more subtle. So this equation that we had back here, where was that equation? This one. This equation ignores a repeat in the genome. If you have repeats in the genome, your Jacquard is also going to change because of the repeat, not because of mutations. So if you know how repeated the genome is, you have to update this equation to take into account repeat parameters as well. And that's what we are doing. So we are like, when you have only 1X coverage, you can't look for patterns of repeat to like do what macrosatellites do. That's a completely different approach. It's a good approach, but it's a different approach. Here we are just correcting for repeat destruction in this equation. Okay, so another example, this one is for beaked whales from a very recent data set that people published. We just reanalyzed their data. Very nice data set. These are like 19 beaked whale genomes from different countries. We see that they have like almost identical, we don't see much population structure within them. It's almost like a star tree, and there is no geographical clustering. The original paper reported something similar. But we also added to this data set two assemblies of different species of beaked whales that were available. So these two are assembly, the rest are genome skims. Now, you can also, again, compute this like within population diversity here is 0.3%. These comparisons are within the species. Now I want to make a point about these two other gaps, which you guys might find interesting. So there is a gap. So these are comparisons within the species. These are comparisons between two species that are in the same genus. So all these skims are from this genus right there. I'm going to butcher the name, but let's say Mesoplodon or something like that. So this is one genus of beaked whales. We have another species from another species from that genus. When we compare those two together, we get this kind of distribution. And then we have another beaked whale from the same family, but a different genus. When we compare across the genera, we get that distribution, right? So there is a little bit of gap between like taxonomic levels, at least in this database and in data set. And I think in many data sets, there will be if the taxonomic units are defined well. And that's another cool thing that might be useful. Okay, I'm almost done with my talk. I'm going to switch to my presentation, to the tutorial in a second. Any questions so far? Okay, so my last slide here. So let me just recap the process, and then we're going to go and actually do it. So you have a bag of reads, whether it's for query or
can I ask you a question, Siavash?
Yeah, of course. Go ahead.
Yeah, so in the last slide, you mentioned within population genomic diversity. My question is, how many individuals would you need like in a population to do that estimate? Okay,
good question. So I don't have an exact answer for you, but I'll try. So in this case, we have 19. And you see this distribution over here. This average is computed over that distribution. So if you have very high coverage data, you can estimate theta even from one individual, as long as it's diploid, right? But we don't have high coverage, so we can't do it with one individual. To get any estimate, you need at least two, right? If you have two, you get an estimate. But how accurate is that estimate gonna be? Well, just look at the variance here, right? This variance over here shows you kind of like the range of values that you might be getting, right? From here to there, right? Now, most of the pairs, most of the pairs give you actually the correct value or very close to correct value, right? But there are some outliers here, right? So these outliers are probably like related individuals. I'm guessing these, for whatever reason, these have higher distances. This might have been an error of our tool, or they are actually like legitimately more distant. This data set has this one outlier sample. Something has gone wrong with that sample a little bit, I'm guessing, but we are not sure what, and we haven't removed it. So you're gonna have some variance. I would say in a case like this, you wanna have at least like, I don't know, something like 4 individuals to get to something that you have some confidence in.
Okay, thanks.
Of course, the more you have, better, but I don't think you actually need 20. I think fewer would have done here. Okay, so recapping the process, for both query and the reference genomes, you have to do some read cleanup. I'll talk about contamination problems tomorrow. There is like a standard read cleanup stuff that you do with BB tools. I have them in the tutorial at the end. If you have time, we'll look at them, but I'm not gonna actually go through the steps. I just have the commands. Then you estimate coverage and you estimate a sketch. Both of these steps are done by a skimmer. And when you run a skimmer, it does both of them. So internally it runs MASH. So it creates a sketch. We can take a look at it, and it creates your genomic parameters. Remind me if I forget to look at those. We will look at them in a second. Okay, so you have now these genomic parameters and the sketches for a bunch of query, for a query and a bunch of references. You compute the distance between the query and each of the reference, you get a distance vector. And that's as far as we are going to go for now. And then we will do some tutorial, and then we'll come back and go after this what you can do. I have more slides for what we can do after this, but I want to go to the tutorial right now. Okay, so this is where you find the Skimmer software. We're going to do installation right now, and this is where the paper was published. Okay, so let me ask Kamal. So how do we do this? So I know we have a 3-hour time slot. Should we go for 1 hour and a half, some rest, and then another hour, like hour out of 20, or do you guys have a different schedule? Oh, sorry, I think you're on mute.
I'm unmuted now. I wonder if Kamil hasn't unmuted himself. It's up to you.
Sorry, I was searching for the program. So I think we have 2 more hours today.
Yeah.
But
so what I prefer to do, if it's okay for everyone, is to do a little bit of this tutorial, then take a break, do a little bit more tutorial, and then do a little bit more, like do the second half of the tutorial.
But I also forgot to mention that today the last half an hour will be a talk, and tomorrow it's like full 3 hours without anything else.
Okay, got it. Good. So I have to. Okay. So let's do just the installations then. Then we'll take a break. That way, people can finish the installation while we are in the break, and then we'll go through the steps. Okay. So I'm hoping everyone has access to this. So if you go on the wiki pages, yeah, there you go. On the wiki, the one we are looking at is, what's today? Today is a Wednesday. So this is right there is what we are going to be looking at. So yeah, you have access to this. This is a recording of a slightly different version of this tutorial I did like a couple of months ago. If this is recorded, I'm going to update this with with this recording later. This is from a previous one. So we're going to look at the tool Skimmer I just talked about. There are 2 other tools. These are for phylogenetic placement using these distances. I haven't talked about those yet. Hopefully we'll get to them today. Otherwise, I'll just talk about them tomorrow. In the tutorial, so these are tools that we developed. We are also going to use a bunch of tools that other people have developed, and we need to have all of them set up. The papers that I talked about are kind of referenced here. Okay, so let's get down to installation. Now, for today, your installation is actually easy enough because we have installed pretty much everything already on this cluster. But I don't want you to be limited to this cluster. So later on, when you go home and you want to install the stuff on your machine, I have instruction here for how you want to like install the stuff on your own machine, right? So these instructions here are what I follow to kind of do the installations that we have on this cluster. We're going to use the cluster right now, this cluster right there. But at home, try, or maybe even in the break, whenever you want, try the installation on your own machine and see if it works. If not, let me know. Hopefully I can help. Okay, so we're gonna start with this first line that I just get into an interactive node. So this line is just on this cluster, get an interactive node. So I'm logged into the cluster. Actually, you know what? Let me log off. I want to log off and go to login node 1. Don't worry about that. I just prefer login node 1 for a reason that will become clear tomorrow. Okay, so we are in the login node 1. You guys don't need to go login node 1. Don't worry. Okay, so we're going to run this interactive thing to get into an interactive to get into a node. So now we are on a node. We are not on the login node anymore. I just got a node. Let me see how much memory we have. Oh, there's no stuff. Maybe top works. Okay, if I'm reading that correctly, it's either 200 gigabytes or 2 terabytes. So okay, that's plenty enough. Okay, so I'm in my home right now. We don't want to do stuff on home, so so we are going to go to a different location in a second. But first, let's activate our installation. So I've created a Conda environment that has most of the Python tools installed there. So let's go ahead and activate it. I just activated. You guys should try to do the same. By the way, unmute yourself or let me know if you have any questions, or like in the chat, or unmute yourself. I want to make sure everyone is following. Okay, so all the Conda tools should be available. For example, Skmer should be available, and it is. Now there are a bunch of tools that I didn't install because this cluster already had them installed. So I'm going to just load them, right? In a lot of clusters, there's this module thing where you can load pre-installed stuff. I'm going to load FastME. It's a tool that we're going to use later today after the break. I haven't talked about it yet, but we will talk about it. FastME, actually, I'm not going to use today. I'm going to use tomorrow. So I'm not going to load it today, but tomorrow we will load it. Feel free to do it, but this swap thing is going to take a little bit of time, so I'm going to just skip it. We want to make sure that I put a bunch of the tools that are required in this location, and we need to make sure this is part of our path. Environmental variable. For me, it is already part of my path because I put this in my bash profile. So I already have it. I might even have it multiple times. If you don't have it in your bash, I don't know if it's part of your bash or not. If you don't have it, just export it. It's fine. You can just run this command. It will become part of your path. Okay. So there are a bunch of tools that I put in the path guppy. And then there are a bunch of like scripts, like 1 or 2 scripts that we are going to use that I put there, like for example this one, and hopefully you have access to it. Let me know, anyone, if you have access issues. Okay, so these are all the tools we are going to be using. So I'm going to just quickly check that all of them are available on my machine, right? The Skimmer, that helped, I just did, we saw it's available. Apples is the Phylogenetic Placement tool. I haven't talked about yet. I will talk about it in like after the break. But right now, I just want to make sure it's available. And you have to type it without the typo. Okay, let's try again. Run apples. There you go. And you give it a -H to get the help. Okay, that works. There's another tool called Misa. We're going to talk about it tomorrow. That one is also installed. You guys should do what I'm doing right now. Just make sure you have access to all these tools. Feel free to skip FastME. We don't need it today. And then guppy, which also works. Okay, so we are all good. Let's see, what is this? Oh, art. Okay, so art. I'm gonna also try art. There you go. Art also works. So all of the tools are for me now available on my node. So I'm gonna stop here. I'll stick. Oh wait, sorry. Let's do one more thing. So I'm gonna use some example data sets. I have made this data set available to you all. So before we do anything, you have a limitation on your home directory. So we don't want to do stuff on home directory. We are going to go to this directory called user work. Okay, so where is this directory? For me, For me, it's wait, what happened? Oh, for some reason I'm not able to see it. Oh, there you go. Okay. For me, it's over here. For you, it would be similar with your username put over there. Okay, so go to user work. Yours is going to be, of course, different from mine. And what we are going to do is we are going to create some folder for our tutorial. I use this name. Feel free to use whatever name. Is probably easier if you just copy paste. Then all you have to, I don't want everyone to copy these files. I mean, you feel free to copy them into your location if you want. But instead of copying them, I'm going to just create a symlink to them. I'm going to just link to them. And the benefit of just linking to them is that, yeah, we are not using their space on this machine. We're going to basically just read this. So this is a link to the data where I have them. If the symlink doesn't work for you, feel free to copy it. Just do ls genomes, make sure that you see this. So these are the genomes we are going to be using. In fact, we are going to use a subset of these. We're going to use some of these yeast genomes for our test case. Okay, I'm going to stop here. If you are in another machine and you want to get this data set, this is how you would do it. I'm going to stop here and answer whatever questions, problems people have with installation. In the meantime, let's take I don't know, maybe 10 minutes break. What's the time? Let's see. 8. Yeah, I think if we are back by 8:15, we can get a lot done after that. So I'm going to start the rest of the tutorial at 8:15, and I will answer problems about installation in the meantime. Okay, so I'll quickly recap. So we have a bunch of yeast genomes. We are going to use only used one of them. I think we use this Cerevisiae as the query. Pastronius is a known hybrid species. We are also going to remove that for now as a hybrid. Because it's a hybrid, we come back to it tomorrow. We'll talk about what to do with mixed samples, and in this case, like a hybrid species. So these two we are going to remove. Everything else we are going to name as or reference data set. So this is going to be or query. Everything else is going to be reference. So these lines right there, they just create. So I'm going to just run them. I just copy pasted and run them. It's a very. Did that work? I think they worked. Yes. So we just separated out those data set. Those genomes into two groups: non-hybrids and non-hybrids query. So the hybrid one we removed. For now, we're going to come back to it tomorrow. Okay, so this is going to be our query species. Everything else is going to be the reference. Okay, so we looked at 9 hybrids. We saw that we have all these species. So we are going to treat these as a reference species, and we want to create a Skimmer library for it. So just like I showed on my slide here, we want to create this reference library of like n different species using a Skimmer. So how do we do it? Well, it's just one. The actual thing is, well, this was just all data preparation. The actual thing is just one line. A Skimmer reference non-hybrid. This -t, you can ignore it for. I'll tell you what that means. For now, you can even not give it. It's not important. We will come to its meaning like a little bit later today. Okay, so I'm just running a skimmer. It's trying to use all the 77 processors that are available to me. It doesn't actually need those many. So it's gonna actually wind up just using 7 because we have only 7 species. So it's gonna take a minute or two to run. Hope you guys are following along. It's not gonna be that long unless multiple of us are trying to use the same node, and it's and it shouldn't actually taking a little bit longer than expected. Oh, there you go. Okay, it's making progress. It's almost there. It's now using 49, which is just 7 to the power of 9. It's, you know, for every pair, it's using a different processor. There you go. It finished. So it wasn't that slow. It was a little bit slower than when I ran it on the login node yesterday. Okay, so we have the run finished. So let's go ahead and examine what it did and what it created for us. So I'm going to just do ls. You see this new folder here, library. That's what it created for us. It created a library. So Samuel is asking, the input needs to be FNA or FASTQ. It could be either FNA or FASTQ. Our next example in a bit is going to be FASTQ. We are starting with FNA. Either one is fine. Okay, so it created 2 new things: a library over here, which is a folder, and a file. So let's start with the library. So I'm gonna go to library and look at what files I have. So you see for each of the references, there is a folder and there is a config file. So the config file, let's just quickly look at it. Nothing interesting there. It's basically saying what Skmer length was used, in our case 31, what sketch size was used. In a Skimmer, our idea was, you know, like a speed is not as important as accuracy. So we have increased this sketch size to a very large value, like 10 million. If you wanted the analysis to run faster, you can reduce this sketch size. Always, it's going to make it a bit faster. But I'm not going to do that today. Let's go for accuracy. So, I think I have to go reference -help. Yeah, so you can change the sketch size. You can always use Skmer size. Skmer size default 31 is pretty good. You can't. I wouldn't ever increase it. You can decrease it, but not below 21. We have seen sometimes the smaller Skmers give better accuracy. I'll talk about that a little bit later at the end, maybe if I have time. And the -t, we will learn about it later. Okay, so in the config file, it's just basically saying this library was created with these parameters. Okay, we need to remember that because we're going to use this library later. Then within each of these, so let's, for example, look at this, I don't know, paradoxes, for example. Within each of these folders, we have 2 files, just like I promised, right? Remember this figure? I promised each thing is going to have 2 files, genomic parameters and the sketch. Genome parameter, sketch. Okay, so we have that right here. We have, this is the genomic parameters, the dat file, and this is the sketch. So let's start with the dat file. So I'm going to just output that dat file. And I'm pretty sure most of you know this command cat. It just like outputs the contents of the file to the terminal. Okay, so what do we see here? We see in these DAT files, you're going to have 4 values. One of them is genome length, which we estimated. We can estimate the genome length. In this case, it's assembly, so it's a trivial estimation. We just like count how many base pairs we have. It's a very simple thing to do, right, for an assembly. And everything else is meaningless here because it's an assembly. The error rate is 0. There is no error rate, right? Or if there is, like we have no way of knowing it. There is no coverage. It's an assembly, and there is no read length because there's no read. It's an assembly. So everything is kind of empty here. You just have the genome length. Okay, so that's fine. That's not so exciting. It gets more exciting when we get to the actual like skims. But let's just look at the genome length from all of our species. Let me make this look a little bit nicer. Okay, so we have the genome length from all of the species. They are between like, I don't know, 100 meg. Sorry, that's not 100, 10. Between 10 megabase pairs to, let's say, like 11.5 megabase pairs, or almost like 12 megabase pairs. That's the range of length that these species have. They're all the same genus in this example. Okay, and we could, if you wanted to, look at the size of the sketches that we have. The sketches are not super small, like I said, because we use the sketch size of 100 megabase pairs. If we make it, the sketch size smaller, these numbers will also become smaller. Any questions so far? Okay, so let's move on. We looked at the library. We saw there was another output, which was this distance matrix. Let's look at this distance matrix. It's a file that basically just has a distance matrix in it. Let me not do the color. Okay, it's a little bit hard to see here. If I maybe reduce my size, we can see it a bit better. So I'm gonna make things bigger. Okay, there you go. That's a little bit better. You can't read the exact numbers, but at least you can see the format. Okay, so you have a species on top and you have a species on the rows. It's just a matrix, right? Along the diagonal, you have zeros, and everywhere else, you have the distance between the two species, right? Even though this is a genus, the distances are kind of high, right? The closest two things here have, is this the smallest number? I think this is the smallest number we have, 7%. Most of the distances are high, like 15, 16%. So this is the like highly divergent genus. If you were to look at like genera in mammals, the distances wouldn't be this high. They would be like 2-3% usually. But this is yeast, so it's different. Okay, so we have our distance matrix. Let me go back to a higher zoom so that you guys can actually see stuff. Okay, so we looked at the distance matrix. Now, what can we do with this distance matrix? We have the distances between the references. We haven't touched the query yet. Before I go to query, I want to show you something cool that you can do with the distances from the references already. And that's a phylogenetic estimation. You can go ahead and compute a phylogeny of your references. Because my background is in phylogeny, I couldn't resist adding a little bit of phylogenetics here. Sorry about that. Okay, so we have a distance matrix we just saw. Basically, we have a distance ij between every pair of a species I and j. We just looked at this. To get to phylogeny, you need to first correct these distances. This distance correction is like basically this is a formula that was discovered in 1969. So it's before most of us were born. It's basically if you assume a model of sequence evolution, to go from distances the distances that we have is the number of observed mutations. If you want to estimate how many mutations actually happened, the number of mutations that happened is always more than what you observe. And the reason is because you can have back mutations, right? And you can have things like convergent mutations. This equation, under the simplest model where everything is kind of like completely random, base pairs are randoms, everything is random, this equation, Jukes-Cantor, is under the Jukes-Cantor model. This equation converts your genomic distances to phylogenetic distances. So this is the number of mutations that happened. This is the number of mutations that are observed, or not number, but like sort of probability of mutation. Okay, so once you do this transformation, which is called distance correction, you can just take these matrices and give them to distance-based phylogenetic estimation methods like neighbor joining or minimum evolution, and you will get a phylogeny back. The tool, so this is an example, maybe I'll skip it. It's basically like for some Anopheles sequences, we looked at the CO1 marker phylogeny, and we looked at this Skimmer phylogeny. We compared it to Open Tree of Life reference phylogeny. All the red ones are the ones that disagree. And you can see that the CO1 marker phylogeny has a lot more disagreement with the reference phylogeny than this like distance-based one, which disagrees in this one very short branch. Okay, so that's just an example. This is an example of cetaceans phylogeny. So this is, I think, a suborder to all the whales and dolphins and things like that. The entire, like the most distant two things in this data set are 5% difference. These two, they're 5% different, right? So in mammals, like distances are much shorter. And when you look at something like this, you can learn some useful things. So for example, these baleen whales, you see how they are much closer to the root than everything else? It's because they're known to have a like reduced evolutionary rate. The rate of mutation in baleen whales is less than like dolphins, right? So you can see like their distances to the root is almost half. That means the mutation rate is almost half. So you can, with this distance-based phylogenies, you can get some cool stuff. This phylogeny, I just like, it took me literally like 2 hours to download all the genomes from NCBI and build this phylogeny. It matched exactly with the phylogeny that was published in 2019 with a much more complex process. So these distance-based phylogenies are decent. They're not as accurate as alignment-based ones, but they're decent. If you want like the most accurate phylogeny, I would still go with the traditional approach. What tool do I use for distance-based phylogeny? FastME is my favorite tool. We are going to use that right now in our tutorial. Let me just go back to this. So you see this translation right here that goes from number of observed mutations to number of mutations that actually happen. This is what I said I'll come back to. This is what the -T option does in a skimmer, right? So remember when we ran a skimmer, so just, remember this equation. Okay, I'm gonna clear this up and go back to the tutorial now. If I can manage to do that. Okay, there you go. Okay, we almost managed. Okay, so when we ran a skimmer, we gave it this -T option. This -T option will automatically do that Jukes-Cantor transformation for you. So this distance matrix that we have here is already transformed. Someone is typing here and is not on mute. Please, if you're typing and you're not on mute, mute yourself. So let me just put like run a skimmer again. Reference -help. I think I need a little bit more zoom. These are probably still too small to read. Okay, that should be good enough. Okay, so you see -T apply Jukes-Cantor transformation to distances. Now, distances, if your distance is above 3/4, two random sequences are going to be 75% Hamming distance. If you ever have distances estimated to be more than 75%, that cannot happen under Jacquard model. Under those conditions, it's just going to output 5. So if you see 5, you know, like that's a very large distance in phylogenetic units. You know something has failed. Luckily, we didn't have any of those cases. So because we have already run a skimmer with -t, or distances are already phylogenetically corrected, so we can just go ahead and run FastME on it, which is the next step in our tutorial. Any questions, or should I go ahead?
Hello. There is this model we can use. We cannot use another model, evolutionary model. I mean, it's just Jackson Found.
Yeah. So if you need, if you want to use more complex models, we're going to publish a paper soon about it. Basically, like you can go, ACGT-based pairs are easy to compute, so that can be done as well. We haven't implemented inside the skimmer, but you can like do that yourself. That's easy enough. We can go up to 4 parameters. Because they're reads, we don't know which strand they come in. The maximum complexity model we can do is something called TK4 model, which has only 4 parameters. So we can't go to like GTR, for example, because we don't know which strand each read is coming from. If you had a reference genome and you could tell the strand of the reads, then maybe we could do GTR.
Okay, thank you.
No problem. Okay, so so let's try to quickly finish this FastME and then I'll stop. I think there is another. Oh wait, no. I have until 9:30, right? Not 8:30. Is that right?
Yeah, that's correct.
Okay, good. So we have time. Okay.
50 minutes.
Okay, good. So we don't have to rush too much. Okay, so this distance matrix that we have was in that format that you guys saw, right? It was like a matrix with tab delimitation. To be able to run phylogeny on it, we need to like change the format. I have given you guys this script that changed the format. So I'm gonna just run it. It gets like the file that Skimmer provides, and it outputs the file in the right format. This file is part of it's part of that position that we added to our path, and in the installation steps, I have provided a like direct link to it. It's just a script you download. You can just run it. There is nothing fancy going on. It's just changing formats. Okay, so all it does really is, let's just look at this file quickly. All it does really is adds one number on top, this number 7, which basically tells you how many species you have. You know, FastME needs to know that on top of the file for some reason. Okay, so hopefully that transformation worked for everyone. Now we are going to run FastME. We loaded up FastME module earlier on, so it's implemented. You're going to run it, and you see it's like super quick. It's not even a second, right? It's like 0.000 seconds. Like right away it finishes. So that's that. We have now the phylogeny. Yeah, we have the phylogeny, which is right here. Where is it? There you go. That's the phylogeny. So we asked the FastME to output the phylogeny with this like name, right? And what we gave it as input was that like distance matrix. Okay, so we have a phylogeny. We can of course cat it and look at it. This is not the most productive way of looking at phylogenies, but of course you can do it. What I like for looking at phylogenies is a tool called Nuvik Utilities. I hope I gave a link to it. Yeah. So there is a tool called Nuvik Utilities. Actually, my link here is broken. I'm going to fix this right after the tutorials. it's already installed for us here, so you can just go ahead and run it. So let me find my position. Where were we? Right here. So, oops, no. There you go. This is where we were. So we're gonna just look at it has a command called NewWik display that is going to show, like, just draw the phylogeny for me on the terminal. If you're like me and you like to do everything in terminal, this is going to be a nice way to look at it. So we got our reference phylogeny here, and that's that. Any questions so far? Any questions? Oops.
It says that we violate some triangle. Sorry. Hi. And if you run the ME3 thing, it says that we violate some triangular inequality. Is that something to worry about, or what does that mean basically?
Right. So we can, there is, like, it has a correction using -q that we can run and see for 3 changes. I doubt it will change much. So let me just make sure we're run a skimmer. Did we give it that -t or before that? Let me just make sure. I hadn't noticed that triangle inequality. Where did we run this skimmer? Okay, one second, I'm trying to find. Oh, there you go. Okay, we're close. There you go. There you go. Yeah, we did give it -t. Okay, good. Yeah, so let's run it once with -q, see if it changes. I'm actually, I haven't looked at it, so I'm gonna. I'll tell you in a second what it means. So what triangle inequality means is that for any two, so take 3 species, the distance from A to B plus the distance from B to C, is it always bigger than the distance from A to C? If that's the triangle inequality. And FastME expects that to be the case. When it's not, it can give you a warning. So what we are going to do now is look at the new tree that we got and compare it to the old one and see if anything changed. Okay, so I have to make this longer so that we can compare this to, and I have to make it smaller so that they both fit. Okay, so let me do this one more time. So this is the with triangle inequality. This is without.
I think it still gave the same warning when you compute it.
But then it said it corrected it, right? So let me go up. It says that we, so.
Okay, sorry.
Let me go back up, yeah. Triangle violation, correcting.
Okay. Yeah, sorry.
Yeah, but the distances, wait, so or scales are not exactly the same. So the distance, like the phylogeny itself didn't change, right? The topology didn't change. The distances changed a little bit. So for example, this seems a bit longer than this one. But in this case, the actual topology didn't change. Yeah, I wouldn't when the distances are so large, these are pretty large distances. Remember I told you, like, a skimmer like the most distant two things here are, let's look at our distance matrix. So our distance matrix is this one. The maximum number here is something like, just eyeballing it, something like 17%. Maybe there is something more as well. These are getting close to the limits of what the skimmer can handle. So like 25% is the limit, right? When you get close to there, the accuracy goes down a little bit. And I think the lack of triangle inequality here is because like very little the estimation error in distances can make your like it can violate triangle inequality. But I wouldn't worry about it too much. What I would worry about is if you look at this distance matrix and you see numbers that are like close to 25%, then I would start to worry a little bit. Okay, any other questions? That was a good question. So let's keep moving on. So we looked at the phylogeny. Now here in this example, we inferred a tree from scratch, right? So we came up with our own, like we used the skimming data to infer a tree. Oftentimes, you already have a tree that you like, maybe from a publication, maybe from your earlier work, from whatever. You already have a tree that you like, so you can always use that. But even if you have the tree that you like, for the following steps that I'm going to talk about, you want to always re-estimate branch lengths with the correct unit, which is in this like juice counter units. So let's assume you have a tree. I have like written this tree here in Nuvic format. So I'm gonna just create like a file that has that tree in Nuvic format. I'll call it just backbone. Let's just look at it. Backbone.tree. You can see there is no branch lengths on it, right? Everything is the same length. Before I can do anything else, I need to estimate branch lengths on this tree. So I can use FastME for that as well. So I'm going to run this command over here. This takes as input my distance matrix, but it also takes as this -u option. This -u option, oops, sorry, this should be backbone.tree. Yeah. So this -u option, what it does is it takes my backbone tree that I have without a topology, and it just estimates branch length on it. So the topology is fixed, and so it's going to output this other file that we can also look at quickly. There it is. So now the backbone tree that I had has like actual branch length on it, right? So you can always do that as well. So now I'm going to go to sample identification, query. We haven't talked about query yet. Now I'm going to talk about query. Any questions yet? Let me see, there is a question. I think, yeah, so FastME allows you to use other, so if all you have are distances, like Hamming distances, you can't do anything other than JuxScanner. For more complex model, you need other information, like for example, the ratio of ACGTs. If you have those, you can run a skimmer without -t, and I believe FastME enables correction for those. But like here, we use the correction of a skimmer. We didn't have to. FastME can do the correction for us as well with the right command. You have to look at the help of FastME and figure out the right option. Okay, so all good questions. Let's move ahead. Okay, so now we are gonna do something with the query. So remember, we created this directory with the query file. So what I want to now do is add this query to this backbone tree. I haven't talked about the algorithms yet, but I have at least talked about the distance calculation part. So let's first run a skimmer with a new command. So previously, when we ran a skimmer, the command we use, so let's, so the command we previously used to run a skimmer was this one, right? And what is it? It's saying reference. We ran a skimmer in the reference mode. So now we want to run a skimmer in the query mode. So this is the command we are going to run. Oops. Oh, and then copy paste. So maybe I'll just type it so that you can see. Query, and then we can always just give it -t for juice counter correction or not. We are going to use it. We're going to give it the query file, which is this, again, FNA file. We will move on to FQ later. And we give it the reference library. We run this. It's going to now compute the distance between this query and everything in the library. It's going to take a minute or two. Again, for some reason, things are slower here. So while it's running, let me move on to the rest of the theory. I think actually it's gonna, yeah, it finished fast. Okay, so let's look at what it generated for us. It generated for us a new distance file. And this distance file is now just giving you the distance from this query that we had, right? Or query was this surveys to all of the backbone sequences, right? So just like eyeballing it, the closest species is this paradoxus with 10% distance. So right away, we know that our novel, it doesn't belong to any of the species that we have. It's in the same genus, but it doesn't belong to any of those species. Why? Because it has like 10% distance to the closest thing in the reference library. So keep that in mind, a novel query that is not in our reference. But we got the distances. If there's no questions, I'll go back here and continue our discussions. Okay, I see no questions, so I'll continue. So here is how far we got. We got this distance vector from query to all of the references. Now, what do we do with it? If one of these numbers is super small, like here, like 1%, or maybe less than 1%, maybe you can assume your sample belongs to the same species and call it a day. But that's kind of like unsatisfying. And what if also there is nothing close, like we just saw, nothing was close to our query? What do you do next? Well, if you also have a reference phylogeny, which we just estimated, what you can do is a concept called phylogenetic placement. So what that does is it takes this distance vector as input, and it takes our reference phylogeny as input, and it will add the query to the reference phylogeny, like you see here. So we are going to do that in a second. Before I do it, though, let's understand what this phylogenetic placement does, like under the hood. So the tool that we're going to use is called apples. It's a method we developed. So let's try to understand why do we need phylogenetic placement and how does it work? So part of the why we need it, we just saw. If you have something that is completely new, you have to find where to put it in the tree. But why can't you just put it as sister to the closest species, right? So like in our example that we were seeing in the tutorial, can I just put it as sister in to this closest species? That actually works like a lot of times, but it's not the right thing to do conceptually. Why? Because the closest thing in terms of distance is not always closest thing in terms of phylogeny. Just remember this tree. It's sometimes called Felsenstein zone tree. Let's say this is your query and ABC is your reference. The closest species to it in terms of the distance is going to be B. So just like this distance right here is the smallest distance. So it's smaller than the distance to C. But like the closest species phylogenetically is C. Like the true phylogeny puts the Q as sister to C, but if you just choose the closest species, you're going to go with B. Why do we get phylogenies that look weird like this? Well, because rates of evolution can change. So let's say Q and B both belong to a species that have lower rate of evolution, and A and C have higher rate of evolution. This is why you don't want to always just add to the closest match. Okay, so you want to do phylogenetic placement. How do we do it? We saw we have this query vector that we saw. Now, if I assume the query is added to any specific location in the phylogeny with any specific like distance to the leaves and a specific pendant edge length here, so this is like a placement tree. If I specify any placement tree, I can also compute the distance of the query to the reference taxa using this tree. So these are tree-based distances. Sorry, my notation has changed a little bit here. These are my tree-based distances. These are my sequence-based distances. If the ideal placement is a placement where these two vectors are identical. That ideal placement doesn't always exist, but we can always try to minimize the difference between the distances implied by your placement and the distances that you got from your data. This is called the mean, like least squared error placement. And that is what the method Apples does. It turns out you can find this like optimal placement very, very quickly. Like there is an algorithm we designed that can do this like super fast. So you don't have to look at every possible position and every possible branch test. You can optimize this in a different way. But this is what Apple is going to output for you, the position that minimizes the difference between these two vectors. This is your data, this is the placement distances. If you do this, so we looked at the accuracy of these placements on 3 data sets: lice, genome skims, mosquitoes, like real genome skim, mosquitoes and fruit fly Drosophilas, we simulated genome skims. And you can see that so we just did live on, we had a reference phylogeny, we did leave one out. We removed one species and tried to like put it back, either using apples or using closest match. And you can see that with apples, you have far fewer cases where we get the wrong placement. So this is the percentage of times where the placement is wrong. Now, for the Drosophila, you see 29% of the time the placement is wrong. You might get worried, but don't worry as much. So this is the maximum edge error. In other words, when it's wrong, in these cases, when it's wrong, how many branches away is it? For apples, it's never, in these test cases, it's never more than 1 branch away. If you do use closest, you can be as far as 7 branches away, because for this lice data set, there is quite a bit of rate variation. Okay, so hopefully this convinces you that a better way to use distances is phylogenetic placement instead of just looking at the closest match. Closest match works fine if you have a very good match in your reference data set. The method Apple is also part of, this is where you can find the paper, how to install it, the GitHub. You installed it already for you. So let's go ahead and use Apple. Any question before we actually use Apple?
I have a question. So you would prefer to use these methods in case, like, you cannot generate your reference phylogeny again with the new sample, if they come from different type of data set or if different markers, or you just cannot generate them again together, right?
Very good point. Yeah. So maybe I should mention that. Yeah. So you could, if all. So see what we got from this query run that we ran here was just a vector, right? So I only computed here the distance from my query to references. And with this vector as input, you can just place it, right? But of course, what you could have done was add the query to your reference set, compute all the distances, and with that, we would be able to compute a de novo phylogeny with the query added. So why would you not do that? Well, there are two reasons. First of all, that's a valid thing to do, but if there are two reasons not to do it. One is what exactly you mentioned. Sorry, I didn't catch your name.
Flavia.
Fabio. That's one of the reasons, right? If your reference phylogeny comes from a different gene, it doesn't come from the same type of data, you can't just update it like you said. Another reason could be speed, right? So now in these cases, like the speed is not a problem, right? But if you have super large backbones, like let's say tens of thousands of species in it, and I know like no one has that right now, but eventually we're gonna get there. Then you don't want to like re-estimate the tree every time. You want to just update. Otherwise, we can do what you said. In fact, as luck have it, our next step is very much related to what you mentioned. So let's look at it. Okay, so what if we want to do? What if we want to add the query to our reference set instead of if you want to just include the query as part of our reference library, we could do that as well, just like we were discussing. So let's look at the library. It does not have this servicia in it right now. You don't have servicia in it. When you run this query command, you can always add this -a parameter. If you add -a parameter, it will do what Flavia was suggesting. It will kind of include the query as part of your reference set. And now we are going to get a distance matrix. Let's look at it. We can now get a distance matrix that includes this query in it as well. Or we can always just take this and add it to our distance matrix. But our library right now includes servicia. So I can just I think run a skimmer distance. Oops, distance. Okay, sorry, I forgot to give it the library. Give it this updated library, and now we are going to have an 8x8 matrix instead of 7x7. That's why it's using all 64 processes. It's going to take a second. But this is what Flavia was suggesting. Do a de novo analysis. It wasn't part of my tutorial, but maybe I should add it. It's a good idea. Okay. So once this this finished. So let's look at the distance matrix. Now it has servicia in it as well. See, now you have servicia. You can do a de novo phylogeny of everything that will have servicia. But let's say you don't want to do de novo phylogeny. What can you do? Well, you could do apples. So this is how you will. Okay, so before you run apple, again, there is a little bit of formatting, transformation that we have to do, like always. So there is this. Sorry, I skipped this step. There is this conversion script that we have also created and given to you guys. A direct link is in the tutorials in the installation step. So this just changes this just does a conversion. So like this is what the skimmer outputs. The format that Apple needs is like the other way around. It's like a skimmer outputs a column. Apple needs a row. We just make the column to a row. So nothing fancy going on. So no two tools use the same format, unfortunately. But we have these little scripts that do the change. Okay, so now we are ready to run Apple. So let's go ahead and do that. Run apples. Okay, that was fast. So let's see what we did. We gave it a backbone tree. This is the tree that we inferred earlier with FastME. We gave it this distance vector, this like row distance vector that we just got from a skimmer with the query option as well. And we gave it an output file. What's this output file? It's a file called, it's a JPlace file. JPlace is a format. I have a link here to a paper that describes what this format is. Let us just quickly familiarize ourselves with this format. It's a JSON file. You have the backbone tree here, right at the bottom. This backbone tree assigns a number to each edge, right? So like this is the number for this edge. That's the number for that edge. Like 1 to, is it 10 here? Sorry, 0 to 10. So 11, we have 11 branches. We assign numbers from 0 to 11 to these edges. Then in this case, we had one queries, but for each of our queries, like in this case, their VCA, it tells you where is its placement. Now the format allows multiple placements. Apple right now outputs just one placement. In future, we might update it to give multiple like alternative placements. Right now, it has just one placement. And this is the edge on which it's placing it. So it's placing it as sister to paradoxes. So in this example, the closest match was the best placement. So, you know, that's what it is. Like in this case, closest and best match were not different. And these are basically like how far from the edge, from the parent should be, and how far, sort of what the length of that branch should be, and things like that. Other information like that. So we know where the placement is. Again, this is not the nicest format to look at. So let's do a little bit of format conversion. Remember, I talked about the tool Guppy that we have also installed here and is available for you to download. That tool, let's run it. It takes this JPlace file as input. Is this readable? Should I make this a little bit bigger? Just make it a little bit bigger. Okay. It takes this JPlace file as input, and what it does is outputs a tree file, right? So it converted that JPlace file to a NuVic format file with the new species added onto it. And once we have the NuVic file, we know how to look at it, right? NuVic display. NuVic display would enable us to look at the file. Okay, so where is Servicia placed? Is placed as sister to paradoxes. It shouldn't be too surprising here that the best placement is the same as apples. You know, the rates are not super different here. Like root to tip distances are all close. You get the differences when root to tip distances vary a lot. Okay, so that's our placement with the genomes. Any questions before I move to genome skims? Okay, so
I have. Would this work in a dated phylogeny?
Dated, and so and basically no, right? If you have a dated phylogeny, you still have to do this step that I showed further up right here to convert your branch lenses to like these Jacquard distances. Now we have created another method called depth that works with dated phylogenies, but that one doesn't work with genome skims. You need like actual alignments for that one. So what you can do is transfer the numbers using this command. Translate branch lenses to Jacquard and then place it on that. of course, you lose like the timing information.
Yeah. Okay. Thanks.
Okay, so let's now actually work on genome skims. Yeah, I talked about genome skims so far. I've been using assemblies. The thing is, the entire process remains exactly the same if you have genome skims. So in my data set, I don't, let's simulate genome skims. So one way to simulate genome skims is to use a tool called ART. ART basically takes a genome assembly and creates for you a genome skim. You can tell it what the coverage should be. In this example, we are doing 2x coverage. You could tell it what the read length should be. We are telling it 100. And we are simulating Illumina. So basically, it's going to have error patterns that are similar to Illumina. Don't worry about actually running ART. You can just copy. I've already run it and made the data available. You can run it. In my case, I'm going to just run it so that you see how fast it is, right? So this is going to just run ART and create genome skims for all 8 of our species of interest, right? For each species, it takes like a second or two to create the genome skims. We are creating a directory called skims. It's going to output all these skims inside that, and it's going to again separate out queries from the references. So we are going to now go ahead and take a look at it. Okay, it's finished. So we went into this skims directory, right? We are inside the skims now. And we have again queries, and we have the reference. If I ls both of them, you see that now we have FASTQ files. Instead of FNA files, we have FASTQ files, and we're going to use FASTQ files, right? So let's quickly look at one, for example, the query FASTQ files. Right, so it's a FASTQ file that you should be familiar with all that entails. Okay, so just to make sure everyone understands, sorry, I'm repeating myself, but our references are also FASTQ files, right? References are FASTQ, so genome skims and queries also FASTQ. Okay, so what we are going to do next is repeat basically same exact steps as we did before, except this time we are going to give it genome skims as input. Nothing changes in terms of the commands. So this is the command that we used for building the reference library. This is the one that is going to take the longest, hopefully not too long. I think it takes maybe 2 minutes. Then once that's done, so we're waiting for that to finish, but once that's done, we're going to look at it and see our estimates of coverage and things like that, right? So let me go back to this little figure we had over here. Yeah, so we're going to look at actual coverage values and things like that for all of our references. So all these coverage data. While that skimming thing is running, let me show you my last slide here. We published very recently, well, not very recently, but last year, we published this paper in Molecular Ecology. It's like one of these opinion papers where basically we'll lay out how we think about genome skimming and why we think it's very useful for sample identification and why we think it's underutilized. So, feel free to have a look at this paper. A lot of sort of more high-level questions about how this would be used on the biological side might be addressed here. Okay, let's see how far a skimmer is. Okay, so this is usually the slowest step. So we're getting there. I think this should finish in a minute or two. All the publications I mentioned here are also in these slides. I'm going to make these slides available on the wiki after the talk today. Okay, so let's go back to our, any questions while this is running? Any questions? Okay, so we'll give it a minute. I'll have a sip of coffee while this is running. So the first step was estimating coverages. It used all the processes that are processors that are available. Then it's sketched each of the, so this is the MASH sketching step for computing Jacquard. And at the end, it just 49 here is just 7 to the power of 2, right? So it's just like comparing all pairwise. And then it just outputs them. Okay, so exactly like before, we got a reference distance matrix and we got a library. And now our library is a little bit more exciting. So let's look at the library a little bit more. So what I'm gonna do is, first, like just look at one of these DAT files that we get back from this library. Okay, so remember everything was NA and nothing exciting was happening. Now it's much more interesting. So a Skimmer estimated coverage for us. Remember when I ran ART, I gave it 2x coverage. It's estimating coverage as 2.07. So it's slightly overestimating coverage, very slightly, but 7% overestimating of coverage. It's estimating your genome length. It's estimating error rate. The default Illumina profile of ART that we use that's actually its error rate. So error rate is correct. And read lengths, it doesn't need to estimate. It's just like, it looks at the reads and sees they are 100 base pairs, so it just puts it in there. So it did a pretty good job, not a perfect job, but a pretty good job of estimating the coverage here. Now, what we can do, of course, is look at this for all of our species. And let me make this a bit larger. So for everything here, you can see the coverage, length, error. And the coverages, all of them should be exactly 2, right? But all of them are slightly overestimated. Sometimes 2.1, sometimes 2.01. Very slight overestimation of coverage everywhere. If someone was interested to know why, ask and I'll answer. If no one is interested, I won't bother you. Okay, so we have these parameters that were estimated. Now, coverage and error rate is limited to like skimming data. FastME files don't have it. But genome lengths we also have from actual assembly, right? So what we can do is compare the genome length that we get from the genome scheme and what we get from assembly. And if everything goes like perfectly fine, those numbers would match. When they don't match, we have a little bit of error, right? Actually, you know what? We'll do that in a second. That's our next step. So before comparing these genome lengths to the assembly, let's do this. Let's also now add the query to the reference library. So the query is also now a genome scheme, a FastQ file, right? And we are going to add it to our reference library. So remember this -A option meant that, just insert this to the reference library. So next time is going to be part of the reference library. Okay, it's going to take a couple of seconds. Are there plans to create a huge reference type database from NCBI data? So that's an interesting suggestion, Ronald. We think a better way to do it is to create pair, like a library pair group. Like you don't want a database that has both insects and plants, right? If you have a sample, you probably know whether it's an insect or plant. You don't know which is specific like insect, right? So it has to be, I think, somewhat categorized. We would like to do something like that. Eventually, that requires a lot of resources, computational resources. We don't have that right now, but we would love to sort of partner with someone and create something like that. Okay, so the library is added. Let's look at the distance matrix. So when you look at the distance matrix, it has just like .fna things in its name. I don't like those. I like to remove them. So let me just remove those. So this command does nothing fancy. It just removes those .fnas from the names. Okay, so now we don't have those .fnas. That's just like aesthetically not pleasing. Okay, good. Now we do the same kind of format conversion that we did before. So make this column thing and translate it to a row vector. So we have a row vector of the distances from Cerevisiae to all of the references based on the skims. So now that we have run everything for skims, let's compare the results that we got from skims from those that we got from the assembly, and see how that looks like. So I have a bunch of commands here that do those comparisons for you in a nice enough format. So don't worry about the details of these commands. Let me just make things smaller so that we can see that everything. There you go. Okay, so column-t, oops. There you go. Okay, so I can make this a little bit bigger. So the stuff on the right-hand side, sorry, I couldn't make it bigger. The stuff on the right-hand side, these are genome lengths estimated from the assembly. Left-hand side is from the FastQs, from the Genome Skimming 2x coverage. So let's just compare a bunch of them. Say the font is small. If I make it bigger, you can see all of them. And these are pretty good estimates, right? So the true length is 11.39. We estimate it to be 11.42. Not too bad, right? It's 12.1. We estimate it to be 11.8. So the genome lengths, as if you just look at all of them, they are all very much in the right ballpark. And again, we are estimating these from skims, so not bad, and just 2x coverage. Okay, so that's the first comparison I wanted to do. Then we want to also compare the actual distances. Column B. Never. Yes. Okay, so these, again, on the left-hand side, we have the distances of the query to references based on skimming data. On the right-hand side, we have the distances based on assembly. So assembly, you can think of it as the gold standard, right? And again, these are pretty good estimates, right? So usually it's most important to get the smaller distances correctly than the long distances. This is just because of how distance-based phylogenetics work. We kind of discount long distances. I didn't discuss that, but that's part of distance-based phylogenetic methods. So the smallest distance is very accurate. The other ones are also pretty accurate, right? So I think this one I can make a little bit longer and you can still see it. If you look at these numbers, they're pretty good. Sometimes when the distances are high, we underestimate them a little bit, right? And we saw that in our simulations in the beginning as well. But for the shorter distances, they're pretty good. Okay, so skimming worked basically here in terms of getting good distances. And now we can go ahead and use apples to add this skim to the reference phylogeny. exactly same command as before. Nothing has happened. Say I'm repeating the same steps as before, just with the skims. And now we can look at this tree, and we want to compare it to the previous tree to see if there is any difference. So this is the tree that we get from skimming data on top. On the bottom is going to be the tree that we got previously. This is like previous directory, right? From the reference, from the assemblies. Let's compare them. That doesn't fit the page, so I need to make it smaller. Let's compare them and see if they look good. Okay, so the topologies are identical. Service is the one that we are placing, right? So the one on the top is based on genome skims. Look at this branch length, this is slightly different, right? So here it's a little bit shorter than Paradoxus. If you had assemblies, you would see that it's a little bit longer, but you know, these are like very small differences. Other than that, we got the right placement, and the branch length is also pretty correct. So the genome skimming worked perfectly fine in this case, even though some of the distances were slightly underestimated. Okay, that's all I had that I wanted to cover before, like in the morning today. So there are a bunch of other points that I have in my tutorial. We don't have time to go through those, and I wasn't even planning. So basically, the first part is, you know, reads need to be cleaned up a little bit. We do that using BB tools. And you can use whatever tool you like. I have put here all the steps that we use for kind of like merging reads and cleaning up reads and removing adapters and things like that. That's the first point of what I was not going to go through. The second part is, like, we used Apple here for genome skimming, but Apples, you can also use it for phylogenetic placement when you have alignments. That's kind of irrelevant to our topic today, so I'm not going to go through it. But I put here some some instructions if you were interested to use Apples for alignment-based phylogenetic placement. I'm not going to go through that. So let's stop here. If there are any questions, I'll answer. I think we have very little time left before the next talk.
I am actually interested in why you slightly overestimate the coverage with.
Okay, so because we don't have too much time, very quickly, the answer is repeats. So the point is, if your true genome has repeats, we are not modeling repeats in normal Skmer. Tomorrow I'll talk about respect which models repeats. If you have repeats, it kind of looks like you have a little bit more coverage because some parts of the genome are exactly the same, repeated twice. You kind of see it twice, you think the coverage is higher than it is.
Yeah, that makes sense.
So that's kind of like intuition behind it. And tomorrow we'll estimate actually the repetitiveness of these genomes, and you will see that it kind of matches up the numbers we were seeing.
Thanks.
Okay, any other questions? Okay, let's stop. We have a quick, I think Kamal mentioned we want a very quick 5 minutes break before